Welcome back to AAKP's annual Policy Summit. This year, we're very focused on government determinants of health, and we're walking through several different issues with our distinguished guests that focus on various drugs, devices, and diagnostics and where they stand on the federal landscape and the importance they have, not just to patients, but to researchers, practitioners in the private sector. Our next panel is entitled Opportunities and Barriers in the Advancement of the Next Generation of Organs, How the U.S. Food and Drug Administration Decisions Impact People Living with Kidney Disease and Kidney Failure. And on this panel, it's very important for us to understand where we stand in the United States right now in terms of xenotransplantation. There have been a lot of news stories that are highly optimistic, but to get to the point where xenotransplantation is a large-scale replacement therapy for patients, it's going to take a lot of work and a tremendous amount of engagement. And so we have two experts who can shed light on this issue. It's captured the public imagination, and we've had very, very brave patients who've gone through a series of xenotransplants for research and for study, and we honor their efforts, and we honor the doctors who have worked with them and the researchers. Our first guest is Dr. Winfred Williams. Dr. Williams is from Harvard, very well known in this topic area. He's the deputy editor of the New England Journal of Medicine and considered a real pioneer in the efforts not simply to advance the science of xenotransplantation, but also as a patient advocate, he understands the urgency that we feel as patients when we're suffering organ failure and our options are relatively limited, especially those who are on the organ waiting list. Our second guest will be our very, very good friend out of Chicago. Kevin Fowler has been a longtime patient advocate. He is the CEO and founder of The Voice of the Patient. And we've worked with Kevin for over a decade. As a fellow patient, he's been a board member of AAKP. He's been very involved in the Polycystic Kidney Disease Foundation. His family has a history in it. He has a background in industry, in the pharmaceutical industry. He's been a close ally on public policy. But probably most importantly, Kevin understands and have a very deep, deep sense of empathy for those who suffer. And so with Dr. Williams and with Kevin Fowler, I'd like you to hear where we stand on xenotransplantation. And with this, why don't we go ahead over to you, Dr. Williams, and you can start.
Great. Thank you very much, Paul, for that introduction. I just want to say that I'm happy to be here at the 7th annual AAKP Public Policy Forum, and I really appreciate the remarks of our previous panel, my good colleague and friend Ken Newell, and PhD candidate Jonathan. Thank you very much for your remarks. And thank you to Diana Kleins and the CEO of AAKP for the kind invitation to be here with you this morning. So I'm going to turn the page a bit, turn your attention to what we feel is an exciting new frontier in transplant medicine, and that's the promise and potential of xenotransplantation. That's the transplantation of an animal organ into a human suffering from end-stage kidney disease. And I don't have to convince you of the need to move innovation forward. We have some 500,000 patients on the end-stage kidney disease dialysis rolls in the United States. Fully, only about a third of those patients, 25%, get kidney transplants yearly. There's a huge supply-demand imbalance in terms of the organs available for transplantation. And the net result of this is that our patients are suffering. Some 17 patients die each day on dialysis, waiting on the dialysis, on the National Kidney Wait List for transplantation. So this is very exciting work that we'd like to present to you. This is the world's first gene-edited pig kidney donor to live human xenotransplantation that was performed at Mass General on March 16th of this year. I'm Dr. Wynn Williams. I'm a transplant nephrologist at Mass General. I'm the Associate Chief of the Renal Division at MGH. And I'm a deputy editor at the New England Journal of Medicine. no disclosures. So what is the opportunity here? The opportunity is to be able to have the capacity on demand to take an organ off the shelf and implant that organ, transplant that organ on demand for a patient in need, suffering from advanced chronic kidney disease, end-stage renal disease. These organs are normal from healthy, genetically engineered pigs. The technology that we've used to undergird this technology is generated by eGenesis, a company in Cambridge, Mass., in close collaboration with researchers at Mass General. So these organs have defined quality, no brain death, no potential transmission of disease, we think, because they are grown in pathogen-free facilities to minimize infectious risks, and the genetic engineering has inactivated important porcine-mediated genomic elements that can confer a zoonotic disease transmission, infection, porcine endogenous retroviruses that have been inactivated in the pig genome. And so the potential here is an unlimited supply of kidneys if this technology is successful. What are the barriers? Defining a donor species, and so we have now focused on the pig, not a non-human primate, because of its breeding characteristics. We eliminated the potential for hyperacute rejection in this model. Humans have natural antibodies against pig glycocarbohydrate antigens on their tissue surfaces, so if you implant a pig that's not been genetically modified a pig kidney, then you will reject that kidney via hyperacute rejection process within minutes. I'm going to show you the decedent model. This is an intermediate step between the non-human primate and a live human subject recipient. This is courtesy of Bob Montgomery at NYU, who produced this kidney model. And you can see that the kidney has been implanted, this is a pig kidney, their model, on the inner thigh of this deceased patient. And you can see now that you have access, immediate access, to urine output, blood flow, tissue acquisition by biopsy. You can measure the urinary chemical components at will in a protocolized fashion. This was their first successful pig to decedent human kidney transplant. Again, this is an individual who's brain dead with a GAL, this is an alpha-GAL knockout, one of the tissue antigens I spoke of in the pig kidney. 54 hours after post-reperfusion, after implantation, you can see on the right panel, that kidney looks really quite good, well perfused, pink, and really quite healthy. The second one they did in November of that year, 2021, and these results were published in the New England Journal in 2022. And you can see that the kidney at 54 hours post-implantation looks quite robust. And this is their GAL-safe, alpha-GAL knockout pig kidney. And if you look at histology at 54 hours at the termination of that experiment, you can see that, for those of you who know histology, that the glomeruli are normal. That's a glomerulus in the center of that right panel. There's no evidence of microvascular inflammation, and the kidney structures, I won't get too technical with you, are really quite healthy, and there's no demonstrable infiltration by lymphocytes. So this is a healthy looking kidney at 54 hours. There are also important ethical issues and challenges that beset this work. And just to review them really very quickly, we want to optimize the safety of this transplant to an experimental subject to avoid predictable adverse outcomes, for example, the transmission of zoonotic disease from the animal vector to a human recipient. So there's a direct risk of symptomatic and even asymptomatic infection that we're trying to mitigate. For the patient's family, friends, close contacts, and the community, the same risks obtains that there could be transmission from an animal vector to the patient's close contacts in the community. And then there's the subtle risk of stigmatization and ostracization, that you've got this new found kidney, this innovation is coming from a pig, are people going to be comfortable with that individual? And then finally, for society, again, to mitigate the risk of potential pandemic disease for the public health writ large as a result of this model. The concern here is zoonotic transmission of infection from an animal vector. So let me tell you about our patient, who is an amazingly brave individual. This was Richard Slayman. His name is well known in the national and international news. He was a 62-year-old African-American man, my patient with end-stage renal disease from type 2 diabetes mellitus and hypertension. He had diabetes for 30 years. He was on dialysis for 7 years until he received a kidney transplant, a human kidney, in 2018. And then over the next 5 years, his kidney began to fail. He had a biopsy in 2021 that showed early recurrent diabetic nephropathy and what we call focal glomerulosclerosis, injury of the nephron elements in the kidney. And that kidney eventually failed. He returned to dialysis in 2023. And then in the year between 23 and 24, his dialysis course was just fraught with recurrent vascular access failure, multiple interventional radiology intervention, vascular surgery, and we could not find a successful access for his blood circulation for the application of dialysis. He was very frustrated after that year. He said, "Doc, I just can't go on like this. I don't want to go on like this." And he was considering withdrawing from dialysis because his quality of life was just so severely deteriorated. So we looked at, via the SRTR calculator, he had a 16% chance of getting transplanted in the next 5 years, and we thought he would not survive dialysis for another year, given his access difficulty, and then we approached him as to be the first patient in our trial. We used a eGenesis 1784 pig, modified genetically engineered pig. This has three gene knockouts. I'm not going to get too technical here with you, but the alpha-gal is knocked out along with other glycan androgens. There are human transgenes that are also edited into the pig genome to increase compatibility with the coagulation, inflammation, innate immunity, and complement systems in humans. And then there are 59 CRISPR-Cas9 gene edits that are accessed to inactivate 59 porcine endogenous retroviral genetic elements. So the transmission of PERV, porcine endogenous retroviruses, is inactivated in this animal. This was our patient in the OR on a Saturday, March 16th. This is made the front page of the New York Times. The Friday after, the 62-year-old man continues to recover after receiving a kidney from a genetically engineered pig. This was the kidney 5 minutes after implantation. We're in the OR. It was just an amazing moment. The kidney perfused immediately and began to produce urine on the table. This is a picture of kidney function, and many of you know that the biomarker we use routinely is serum creatinine. This creatinine reduced to baseline within the first week, now with a baseline creatinine of about 1.5, 1.8. He had a rejection episode on day 8, and what's interesting about this is that when we worry about antibody-mediated rejection, that was not the case here. It was just cellular-mediated rejection, garden variety T-cell mediated rejection, easily treated by standard anti-rejection medication, and he recovered good function. His terminal creatinine was 1.5 to 1.8, and he had robust urine output. There was no evidence of transmission of xenopathogens by way of what we have a metagenomic platform using sequencing to detect and quantify microbes in the blood, and an array of viruses as indicated here, all negative at all time points. He was doing reasonably well. He had frequent clinic visits. We were seeing him 3 times a week. On the morning of the 51st post-transplant day, he was evaluated in clinic, reported not drinking well, was a bit fatigued, creatinine was slightly elevated, and ultrasound was unremarkable. We gave him a half-liter of fluid in the clinic. We also infused magnesium to boost his magnesium level. And he spent the rest of the day with his fiancée, going about the usual activities of daily living. They went to the grocery store a couple of times. He still complained of fatigue that day. And later that evening, while he had gone to bed, his fiancée discovered that he was having respiratory distress, and he became unresponsive. EMTs were called. They attempted resuscitation that continued at the nearest adjacent hospital, the nearest hospital they took him to, where I met them, but resuscitation efforts were unsuccessful. We did get permission for an autopsy. That autopsy showed that he probably died from a cardiac event. You can see here that on the left panel, his heart rate was twice that of a normal human heart. He had severe hypertrophy of the heart. He had diffuse multifocal myocardial fibrosis, but there's no evidence of an acute myocardial infarction. We think that he had a cardiac arrhythmic event, given the magnitude of his diffuse ischemic heart disease. There is no evidence of pulmonary embolus, infection, or inflammation. And there is no evidence of rejection or thrombotic microangiopathy. These are entities that we're concerned about in a functioning kidney in this model at day 52. In the autopsy, you can see that there's no evidence of rejection. So what will the FDA require? They will require a consistent pig phenotype. There are about 5 different gene-edited models now in circulation at the major centers of where this work is being done. We need a defined platform of immunosuppression that's routine and protocolized, and then there need to be consistent preclinical results in a life-supporting model before we can move this technology to clinical trial. This is my patient, Richard Slayman, an amazingly brave and courageous individual. I can tell you that we are steadfast in working on this model of transplantation such that his sacrifice will not be in vain. And this is 6 days after he underwent the first genetically engineered pig kidney transplant. This is our team. We were joyful on that day of surgery. So genomic engineering has potential to ensure broad patient access to transplantation by creating a sustainable supply of organs, an organ off the shelf, if you will, and to address patients early in the CKD disease process, even allowing preemptive transplantation where necessary. And this may be a new paradigm for managing end-stage kidney disease to transform organ failure from a deadly disease to one that's imminently treatable. So I'll just finish here the remarkable potential of xenotransplantation to address the critical shortage of human organs has been met with considerable ethical scrutiny, and it's still being scrutinized, raising profound questions and dilemmas that must be thoughtfully addressed. It's incumbent upon us in the scientific community and policymakers to communicate the steps taken to mitigate ethical concerns and provide evidence of rigorous ethical oversight, animal welfare standards applied in the xenotransplantation research. So this is the team. It's a team effort. Tatsuo Kawai and Leo Riella, or the principal investigators on the trial, and a team from eGenesis in Cambridge and others. Thank you very much.
Thank you very much, Dr. Williams. And now what I'd like to do is move to Kevin Fowler. Kevin, you have such a unique background as a patient who proactively got a kidney donation prior to having to go on dialysis. So you did preemptive transplant. You have a background in the pharmaceutical industry. And of course, you've worked very, very closely with FDA to raise patient insights and lived experiences. Can you tell me your reactions to what Dr. Williams has presented and your insights on what the path forward is going to look like and what it's going to take for those who are listening and for federal officials in order to keep the ball moving on this.
Yeah, thanks, Paul, and thanks to AAKP for the invitation. If you guys could pull up my slides, I'm going to build upon what Dr. Williams said. I think the main thing is to take a look at is kind of go back and look at what our problem is that we have right now in the United States. The problem is there's 467,000 people on hemodialysis. There's roughly 13,000 on home, and then I think there's about 67,000. So I think take a look at the problem right now. And Jonathan was talking about this earlier too, is that look at kidney transplants, half fail at 10 years, right? So that list of hemodialysis patients is growing all the time. And so I had a conversation with my transplant nephrologist at Kidney Week, and we were talking about dialysis. And he said, well, and I was trained on it 35 years ago, it hasn't changed. Think about that. And then you have other areas like they have ventricular assist devices in a much smaller population. So I think like what Dr. Williams said, I'd like to speak to is the human suffering on hemodialysis, the desperation that Mr. Slayman spoke about. I saw my mom on dialysis. I've seen many friends die on dialysis. And what I find hard to believe is that we haven't had any advancement in a better treatment for end-stage kidney disease than a dominant form of hemodialysis. It's just unconscionable. So I think if we take a look at that, is that that's the problem. And I think with that as a problem, if we have an opportunity to investigate xenotransplantation, and I also want to point out to artificial kidneys too, right? Going back to the executive order, this is the Advancing American Kidney Health Initiative back in 2019. If you look down at the bottom and look at one of the priorities, it mentions down there catalyze the development of the artificial kidney. So to look at this fairly, there's been a lot of progress on upstream, identifying kidney disease earlier, and adoption of treatments, okay, not great, but there's been a lot of advancement on that end. But if you look at the ESKD market, as Dr. Newell mentioned, there's nothing happening. This is ridiculous. Plain and simple, it's ridiculous. So I think that if you have an opportunity to look at xenotransplantation as a potential to offer maybe a bridge, all the effort should be put into this to investigate this to see whether this is viable, right? I'll be honest, I have a lot of questions on the subject that I'd like to know about, and I think the patients have to be first and foremost involved in this process. But the fact is that we're accepting this list to grow every year and to keep telling ourselves we're doing a good job. The patients aren't saying it. So, I think there's great promise here. I would just make a call to action is that I think the ESKD market, looking at improving kidney transplantation, artificial kidney, how about new dialysis treatments, xenotransplantation, is really being overlooked right now. And I call, a call to action to regulators is to engage with the community and begin to take this issue really seriously to move away from a treatment that hasn't changed since my mom was on it in 1986 when she died. That's my position. Just want to bring this up too, is that there's two things that I follow just in terms of guiding me as principles. And one is this book written about Dr. Scribner by Dr. Christopher Blagg about the early days of dialysis. And I know Dr. Williams, you know this, and Dr. Newell, but in many ways, Dr. Scribner was the prophet who called everything that's happened in kidney disease. He spelled it all out. And so when we're bragging and saying, hey, we're increasing home dialysis, we're only going back to a time and place when that was a common form of treatment. Patient engagement was principle. So I think a lot of ways we go back to what he talked about, and the whole aspect about renal rehabilitation is an unfulfilled promise that has not been delivered upon. So anyway, those are my thoughts.
Thank you very much, Kevin. And you make me smile and you make me sad. It's very important for those who are watching to understand this. In 2016, there was a White House summit on organ donation to take a look at what could be done to increase opportunities for transplantation and to beat the curve of status quo dialysis in the United States. It has a 50% mortality rate. And the path forward on that is innovation in transplantation. You've heard a very powerful case for that by Dr. Williams. You've heard the patient burden side of it from Kevin Fowler. In 2019, there was a presidential executive order on advancing American kidney health 5 years ago. And I guess our point is this. If you can have two presidential administrations, a bipartisan national call to attention and change over the past 8 years, then the time is now to act. And the time is now for FDA to listen to the burden of patients, patient insights, and the needs that industry and researchers have to have clear targets to work towards so that we can save more lives and reduce the cost of the American taxpayer. With that, I'd like to say thank you very much for the past two panels and to our guests.